here is kind of like the second group tackling architecture 5. So please take it away. Your time's.
Yeah, hi. So we decided that because there were so many of us, we'd split into 2 groups. We didn't talk to each other at all. So we've come at the problem from very different points of view. What happens when you put 5 small molecule experimentalists together? They answer a completely different question to everyone else. So it's going to be a little bit different. So we've gone for this catchily titled meta nanosome. And we've got this nice cartoon here. So if you like a cartoon, stick with that for now, because there's some scary chemtrails coming up later. So, you know, we had 2 hours to solve this problem of basically how to do effectively 1D, 3D printing. Now, we cheated and stole biology's homework, because biology's had 3.5 billion years to solve this problem. So what we thought we would do is have a look at biology and think, well, how does it do it? And I want to emphasize what we're not interested in doing is reproducing biology. Biology already does biology fantastically well. What it does do, interestingly, is interesting phenomena. But biology only knows about 23 different reactions. I, even as quite a rubbish synthetic chemist, know a few hundred reactions, right? So there's a whole wealth of chemistry, unexplored space out there that we can start using to create the same phenomena that biology does, but start doing it in a fully artificial manner. So again, as I say, we set ourselves the challenge of not using anything biological and the challenge of making it as small as we possibly can, because, you know, we all like the word nano in front of things, so let's make everything smaller. Okay, so the first question we asked was, we asked ourselves the question, what does the ribosome actually do? And what are the sort of elements that we need if we want to start thinking about how we can do this in an artificial manner? So we need some sort of an information polymer. We need some way of reading that information, which involves directionally moving along this information polymer, and some way of reading whatever the alphabet of code is on that polymer. We need some way then of writing it, i.e. putting an output, translating it. So in the case of a ribosome, it reads your RNA and translates it into a protein. And then, of course, on top of that, what isn't involved in the ribosome itself is this idea of error correction. So that's completely separate machinery that does this, because the ribosome just isn't good enough for biology. It makes too many mistakes. And we as synthetic chemists can't possibly hope to do better than the ribosome, not at the moment anyway. So these are other things that will need to be looked at. This idea of how can we do the error correction, and ultimately also, as we've just heard from the previous group, how can we actually fold these structures up into something useful? Those 2 things are separate and beyond the scope of what we've looked at. So we've looked at the real fundamentals of actually how do we, effectively, if you want, print in 1D. How do we make a sequence programmable polymer? So when we look at sort of the information that's used out there, there's various bits of work out there. I just want to highlight this work here from the group of Chris Hunter over in Cambridge. So what they're doing is they're looking at how you can store information on polymers and how you can potentially then replicate that information. Now, of course, with the ribosome, you're going a step further, because you're not trying to replicate the information. We're not trying to create the information thread we started with. We're trying to transcribe it into something a bit different. So again, we can look at our alphabet, and there's all sorts of chemistry out there we can use. I did warn you there were going to be scary chemdraws. Just the top one here, the imine formation, top left, that's the one that we've decided for this sort of proof of concept design. So this is a very precise way that you can get this dynamic reversible covalent bond formation between an amine and an aldehyde. And this is going to be the basis of our alphabet that we're going to use. So we can think of those as 1s and 0s. We can have aldehydes as 1s, amines as 0s. So we're just going to write in binary for now, because again, it just keeps the whole problem much simpler. And then we've got these other interactions. Some of these are orthogonal, which is going to be very useful for what we actually need from this machine. We then need to think about how we can make a reading head stage. And we saw some beautiful work yesterday from Jim Seal, who's probably somewhere, there he is, showing us about some of these pumps that Fraser Stoddart has developed, where you can move this macrocycle directionally on a polymer. And now, if you remember, one of the things that's absolutely crucial to reading information is to have that processability. The ribosome moves directionally along a polymer, and that in itself is a big problem. So that's an important bit. But we can see that there are solutions out there where we sort of take these sort of pumps that like Fraser's developed, like other groups are looking at, and actually move directionally along the polymer. And in fact, there's an example here from the Lee group, if the video works. How do I click on link? Control?
You just said Yep.
Sorry.
No worries.
If I can't work this technology, I can't hope to make this technology work. So, sorry, yeah, this is an example that was recently published from Dave Lee's lab in Manchester, where you've got exactly that. You've got an information polymer, so we've got some chiral information on that thread, and the macrocycle goes along and responds to the information on the thread, and it reads it out as a CD response. So again, we can just see that there are groups out there sort of trying to address some of the problems that make up the overall bigger problem of making this artificial ribosome. So, Oh, we're back in scrolling land. So we've gone about how we actually think about designing this. And again, so we've come up with this idea of we're going to use macrocycles because that's a good way of ensuring process solidity so that you don't fall off the information thread. And we know that we can pump macrocycles along polymers using techniques like those shown by the startup group. We're going to have some information on our polymer. So our polymer is just going to be comprised variously of aldehydes and amines. And that's going to allow us to read that information. We then need some way of actually bringing in our building blocks. So if you think this is equivalent now to our ribonucleotides that come in. And so what we've done here was we've added these orthogonal groups here. So we can bind here a boronic acid, and we can bind here a thiol. These are, again, reversible reactions. These are going to be coming on and off. But the key point is that we hope that when the macrocycle binds the information polymer will form the imine with the relevant binding site so either here on the left or the right, and that electronic change will then activate the group over here towards a ring opening metaphysis reaction. So ultimately that will then propagate, and we'll be able to use this living polymerization of ring opening metaphysis to build up the polymer in a stepwise way, effectively being able to take any programmed sequence and being able to exchange that out and keep the same machine to read any number of different tapes, if you like. So ultimately, as I say, we're going to scroll through that. Error correction and the actual folding bit, those are two completely separate questions, of course. But if we just think briefly about killer apps, well, I mean, the main one, of course, is we're thinking about information storage here. And this is how biology does information storage and processing. And of course, we should remember that human brains are millions of times more powerful than the very best silicon computings. So clearly, starting to address these problems is going to get us there. And then, of course, we can think about using sort of the AlphaFold approaches that have been so successful, incorporating AI to predicting protein folding to try and use it on these artificial metaproteins, if you will. And just very finally, the £5,000. This is a very difficult proposal. This is requiring expertise from so many different fields. You know, we need expertise in catalysis, we need expertise in motors, we need expertise in dynamic covalent chemistry. So the kind of thing that would be useful is, after a bit of time going away and thinking about the individual little bits of the problem, get together and have another big discussion about it. So that's it. Thanks.
Wonderful. Also for you, questions, comments? Yeah, will you start?
Do you have any, very interesting, do you have any kind of projections of what kind of, let's say in your wildest dreams, you have massive success 10 years from now, what kind of processivities, yields, speed of translation, these kind of performance metrics would you dream of being able to
being realistic, within the next 10 years, if we've done the first two steps of this, i.e. just got the catalysis working, we're doing really, really well. You know, this is a decades-long project. But ultimately, we have the proof of concept, right? We know that a functioning nanotechnology based on this sort of system can exist. It's called biology. So we know that biology, maybe it's not the upper limit, but we certainly know that that is within the limit. So in principle, there's nothing stopping us getting to the sort of rates that biology does.
But also hard to put a projection of time frame.
Oh, hundreds of years.
Okay. All right.
Okay, I
We
think
think
there
for
was
the
one.
future, right?
Will $5,000 be enough?
It'll be enough to have a meeting to discuss where to start.
Put it in the bank. You wait for a couple hundred years.
Oh, we're all in Europe, right? So $5,000.
I guess it's a long way.
Okay, anyone else? I saw one. Yeah, Will, would you mind maybe passing him on the mic? Thank you.
So, sorry, I lost track of what it was you were actually trying to make. I mean, the useful molecules are proteins.
Well,
And so can you do, you were thinking of doing amide bond formation.
of doing what, sorry?
Amide bond formation.
No. So this is the whole thing. We don't want to use the chemistry that biology has because there's no fundamental reason why we should. Right? So I think we've actually used ring opening metaphysis reactions there. But in principle, you know, it's the fundamental principles that matter, not the specific chemistry. So in terms of the useful molecules that we make, realistically, you're not going to make anything useful for a bit. You're going to be doing proof of concept. But once you've proved the concept, then you have the idea to take any information polymer and translate that into creating the sequence that you want. So in the way that we can, with biology at the moment, take any RNA sequence and synthesize the peptide sequence that we want. That's the fundamental thing, and that's clearly important because we're all alive.
Got it.
So in this
Final.
diagram, the brown string is the product that's being made.
Yes, that's right.
And that's coming off.
And the green string is being read.
Yeah, sorry, the figure's not the best.
Does the green string get consumed when it's read? Or
No.
No, it goes right through.
Yep.
Okay, wonderful. Thank you.